Hi, my name is Maria Deutschmann, and today I want to share with you our idea of using a single-cell, multi-omics, heterogeneous multilayer network to infer gene regulatory mechanisms. Our body consists of a lot of cells, and each of the cells carries the same genetic information. We have many cells and many different types of cells. And we know a brain cell is very different than a cell in a muscle. The different cells have cell-specific functions. And cell-specific functioning requires each cell to regulate its genes. Some genes are turned on, and a lot of genes are turned off. Here, I want to give you quickly 4 observations. The first observation is that in order for a gene to be turned on, that is, for a gene to be transcribed, the DNA needs to be accessible. If the DNA is very tightly packed, then the gene cannot be transcribed. Even if the gene is accessible, we observe only low basal rates of gene transcription because it needs much more than the accessibility of the DNA and the presence of the DNA polymerase. There is a whole machinery involved in the transcription of a gene, including transcription factors. And lastly, transcription factors bind to promoters, and they can also bind to enhancer regions. So we always have a direction. Transcription factors regulate gene transcription. We can represent the transcription factor-gene relationships as a network. A network is a mathematical object consisting of nodes and edges. In a gene regulatory network, the nodes represent genes, and the edges represent regulatory mechanisms. Since these networks represent transcription factor-gene relationships, the edges are directed. And since transcription factors are also genes, the direction can be in both ways. Usually, in single-cell studies, transcription data is used to infer gene regulatory networks. But several benchmark studies have shown that tools to infer gene regulatory networks are rather poor in their performance. One of the state-of-the-art tools is Gene3. We applied it to publicly available data of human and mouse embryonic stem cells. On the y-axis, we plotted the number of correctly inferred transcription factor gene regulations. On the x-axis, you see the number of considered top-ranked potential transcription factor gene relationships. The black line represents the results we expect when we would randomly guess. As you can see, Gene3 is worse than random guessing half the time. And it's only much better than random guessing in 1 out of 4 times. So what can we do? I told you that we usually use single-cell transcription data, but the technological advance in the past years allows us to obtain also other single-cell modalities. And there are several tools, for example Pando and Cell Oracle, that allow you to use single-cell transcription data, DNA accessibility data, and known transcription factors together with their motifs. Very briefly, the idea in both is to first have a backbone network, and then in a second step to model the gene expression using either genetic transcription data or genetic transcription data together with DNA accessibility data. In both cases, we obtain a gene regulatory network. And that gene regulatory network shows the predicted transcription factor gene regulations. We also applied it to the 4 data sets. And as you can see, we are better than random guessing in all 4 cases. And that's already good. and we outperformed Gini 3 in 3 out of 4 cases. I told you that both Pando and also CellOracle give you a network consisting of potential transcription factor gene regulations. We hypothesized that we should keep different modalities separate. So we have one network for each modality. A multilayer network is a set of nodes, edges, and layers, and each layer consists of a single network. They are so-called intra-layer edges within each layer, and the layers can also be connected via so-called intra-layer edges. If the nodes within each layer are the same, the multilayer network is homogeneous. We also call it multiplex. If the nodes are different, the multilayer network is heterogeneous. And this is what we envision: a single-cell, multi-omics, heterogeneous, multilayer network that we can then use to infer gene regulatory mechanisms. We can use it to predict transcription factor gene regulations, but since we keep the different modalities within our framework, we may also be able to infer other biological relationships, such as enhancer gene relationships. We are still testing, but in our preliminary results, we outperformed the tools I have shown you so far in the number of correctly predicted transcription factor gene regulations. So indeed, it seems to be very promising, first, to include other data modalities in addition to single-cell transcription data, and second, to keep the different data modalities separate. Another idea that we had was to use modules to reflect biological processes. A module in a network is a set of nodes that is tightly connected to each other. And the nodes of a module are less connected to nodes outside of the module. Since we have a heterogeneous multilayer network, we would like to find a heterogeneous module. A heterogeneous module contains nodes from different layers. And I thought that may be a simple task and that there are already a lot of methods and tools that I can test. I also found a dream challenge, and the dream challenge even had a section specifically for modules in multi-layered networks. But I was rather disappointed. Most of the participants of the Dream Challenge merged the different layers into one single network, and then they detected modules with a tool designed specifically for single-cell networks. The authors of the paper had another strategy. They first determined modules within each layer, and then they merged the results. However, both strategies do not represent what I had in mind. I want to use a method that detects modules within a multilayer network. 2 participants of the Dream Challenge offered such a method, but one of them was designed for homogeneous multilayer networks, so I couldn't apply it. Only one method was left that I could use, since we could potentially use it for our heterogeneous multilayer network. So far, the results we obtain are not very meaningful. Most of the nodes end up in one giant module, and the other nodes are dispersed in many tiny modules. If you have suggestions on how to detect modules in a heterogeneous multilayer network, I'm very looking forward to talk to you. Thank you for your attention. If you would like to see a network popping up on your Twitter now and then, don't hesitate to follow me. I also want to thank Remy, who worked with me in this project, and our supervisor, Laura Cantini. A big thanks to the Student Council Symposium team for organizing this event and letting me present to you my work. Thank you.
Thank you, Ina, for this nice talk. You can write your questions in the Q&A session if you like, or if you like, you can raise your hands and we can give permission to speak. Okay, I guess we have no questions right now and we have to, oh, there is one.
Is it possible to tweak the weight of the different layers in informing the clustering modality? And that's actually what we have in mind. And so far, we didn't find how, except from putting different weights on the intra and interlayer edges. And that's kind of biased then, because some layers have way more edges than other layers. And some in between edges, they are just way more than the others. So that was one strategy we had, but until now, it did not work out as good yet.